Hi, everyone.
Hello.
Hello. Welcome. Happy Thursday. Welcome, welcome, everybody. Do me a favor if you would and type your name into chat. Thank you. Thank you very much. Yeah, type your name into chat for me so I have a record that you were here today. Very good. Thank you. All right. Very good. Just getting everybody into the meeting. Very good. Thank you. Looks good. Thank you very much. All right, very good. So it looks like just about everybody's here. So if you would, just go ahead and turn your camera on for me. You can leave yourself muted when you're not speaking. And again, if you're just coming in, please go ahead and type your nickname or your first name into the chat for me. That's how I have a record that you were here. All right, very good. Okay. So today we're going to be having our first lecture topic, which is the science of biology. And before we get started into that, I'll just ask if anybody has any questions that I can answer from our last meeting together. If you have any questions about the syllabus or the schedule or anything else that has happened since I saw you last, feel free to type it right into chat and I'll see if I can help you with it. Remember that this week, you don't have any homework assignment to do for me, but there is a student survey that I'd like you to complete. That's in the modules section of our Canvas course, and it's in the Start Here module. A lot of you have already completed that survey. Thank you very much. If you haven't done that yet, if you could just get that into me by Friday at midnight, I would appreciate that. Chris Lynn's asking a question about the lockdown browser. What I'll do is I'll pull up our Canvas page in just a second and we'll look at that link, okay? Okay, so I'm gonna do that right now, as a matter of fact. I'm gonna share my screen with you and I'm gonna pull up our Canvas page. Let me clean this up here. So that's what you should be seeing right now on your screen. You should be seeing our Canvas course, INBIO110, and I've clicked over here where it says modules. And these are the modules that are posted so far. There's that student resources module, the Start Here module. Here's that student survey I just mentioned. And then if we come down into week 1, we're right here. Remember, in every week, there's going to be a to-do list. That's just going to be a nice summary for you to have that shows what's coming up in the week for our class. So as you can see, for week 1, in our first meeting, we had that introduction, and then today we're doing this lecture, Science of Biology. There's no lecture homework this week, but we just want to make sure you get that survey in. by Friday at midnight. Okay, so if I go back into modules, the slides are up here for you for today's lecture, Science of Biology. So you'll always have the lecture slides that I'm using during our lectures, whether we're on Zoom or in the classroom. So you don't have to write every single thing down that's on those slides. You can always access them on Canvas. Now I'm going to go over here into this left-hand menu right here. Can you see this? There's actually two menus in Canvas. Remember, there's this, what I call the far left menu. That's where you get onto the dashboard. That's where you get onto the inbox feature that I mentioned that I want you to use when you message me. And then this closer menu, this near left menu, this is where you're going to find the link for Lockdown Browser. All right. Now, if I click on this link, my page is going to look a little bit different from yours because I'm an instructor, but what happens with Lockdown Browser is there are assignments that do not require it, and there are assignments that do require it. Now, right now, you may not see anything listed under Lockdown Browser. You don't see anything listed because so far, nothing that I've posted is going to be using the Lockdown Browser. Does that make sense? As soon as I set up an assignment in Canvas and I set it up so that you have to use the Lockdown Browser to get at it, it will show up on your end. Now, right now, there's nothing in all of the things that I've posted that requires the browser, the Lockdown Browser, which is why you don't see anything on your end.
Okay, because like I just can't, yeah, I just can't see like when it says like home syllabus announcements, I just can't see the Lockdown Browser tab at all.
You don't see the link for it in that menu?
No.
It's not in it on mine either.
Okay, I'm going to double check that after we finish today and make sure, figure that one out. I'm sure there's a simple explanation for that, and I will take care of that. Now, next week, when we get together, I'm going to be posting a practice quiz for Lockdown Browser. And again, I'll make sure that the link is clearly visible to you. And you'll have a chance to play with it a little bit, to take a little silly quiz, you know, that has silly questions in it. But just to make sure that you're comfortable with it and that you're all set up with it. Sorry, that doesn't need to be there. Okay, very good. Yeah, it sounds like, you know what it sounds like? It sounds like it's just hidden from view. There are actually a whole lot of links in Canvas that instructors can add in to a course or not. And we have to go down through this list and mark these links as to either include them or not include them. So I'm sure that's what it is. I'm sure I just need to redo the inclusion for Lockdown Browser. Thank you. Thanks for letting me know. That's an important thing. If you ever can't find something, or if there's something that I've asked you to do and you can't find it, please let me know, because it's usually just a small error on my end. All right, great. Any other questions? Anything else that's come up? All right, very good. All right, so I'm going to pull up our slide set that we're going to be using today, and we'll get started. Remember, you're going to want to take notes for yourself. Just remember, you don't want to try to write every single thing down off the slides, because you don't have to. And frankly, if you try to do that during a regular lecture, you're going to find it difficult. I think the pace of the lectures is just too swift for you to try to write every single thing down off the slide and what I'm saying. It's more important for you to write down the extra things that I'm saying during lecture, and then pull the slides up when you need to study. Okay. Oh, hold on just a sec. Let me do that one more time just to make sure we're all set. Right there. Okay. So what you should be seeing on your screen right now is the title slide for the lecture. And it says Science of Biology. Remember that this meeting today is going to be recorded. And later on today, after it has finished processing, I'm going to be posting it up onto our playlist. So if you need to come back for some reason and look at this, our meeting today, you'll have that video available to you. So the way we're going to approach our lecture topic today is like this. First, we're going to talk about what's known as the biological hierarchy. We're going to talk about how in a multicellular organism like a human being and in other organisms that are single-celled, how the structural components of organisms are just very generally organized into a hierarchy. We're going to talk about the characteristics that all living things share, what defines life on our planet. And then we're going to talk a little bit about scientific experimentation. We're going to talk basically about how science is done, how research is done, experimental research. Again, very generally speaking. We'll talk about the kinds of reasoning that we use in science. We'll talk about the different types of scientific studies that can be conducted. And we'll just do a very quick review of the scientific method. So whenever we talk about living things, we're talking about a range of organisms that go everywhere from a single cell, because there are lots and lots of organisms on our planet that are single-celled organisms, all the way up to multicellular organisms that are very, very complex, such as a human being. And when we look at living things, we can see an organizational hierarchy based upon the very fundamental molecules that all living things contain. So for example, at the very bottom of every living organism is the atom. Every living thing is made up of a collection of elements. We're going to talk more about what elements you find again and again and again in living things. But atoms of each element are going to come together and combine into molecules. And we're going to, from that point, develop more and more and more complex cellular structures. In a eukaryotic organism, like a human being, molecules are going to come together inside of the internal space of a cell and create these little functional compartments that we call organelles. Organelles, if you translate that word, it just means little organs. Inside of every one of your cells, there are these specialized work houses called organelles. And each organelle is tasked with performing a different function for the cell. For example, the nucleus is the organelle that houses the genetic material, the DNA. The mitochondria is the organelle that performs the job of producing ATP. There are endoplasmic reticulum inside of cells. There are Golgi apparatus. There are lysosomes, all different kinds of organelles that each have a different job to do. Only eukaryotic organisms have organelles, however. There is a whole nother type of organism called a prokaryote that doesn't have organelles. So this would not be included on their list of biological hierarchy. The next level of complexity in all living organisms is the cell. And you'll notice that I have cells highlighted here. Cells are fundamental to our definition of life. Cells are the smallest unit of life on planet Earth. In other words, if it's a living thing and it's on planet Earth, it is made of one or more cells. That's a really important point to make, especially during our pandemic. And that's because we need a definition. We need a working definition for what is alive and what is not alive. Sometimes it's very obvious to us, right? I mean, it's very obvious that this desk that I'm working on is not a living thing. It also has atoms in it. It has molecules in it, but it's not cellular. It's not composed of cells, and therefore we don't consider it to be a living thing. Now, I mentioned the pandemic because right now we're all living with the difficult consequences of having a new virus floating around, the SARS-CoV-2 virus, which emerged back in late 2019. Viruses are not cellular. Viruses are actually particles of material, very, very small particles on the nanometer scale. And they're composed of things that we see inside cells, but they are not themselves cellular. A virus is a very simple particle of material. It's a piece of genetic material, either DNA or RNA. And that piece of genetic material is surrounded by a coat or a coating that's made out of protein. Now, I know you've all seen the famous image of the SARS-CoV-2 virus, the one that has the little spikes coming out of it. That image has sure made the rounds over the past 2 years. The SARS-CoV-2 virus is a particle. It's a piece of genetic material that is surrounded by protein. It does not meet the definition of a cell. It doesn't have a couple of things that cells have to have in order to be considered cells. And therefore, this viral particle, the SARS-CoV-2 viral particle, is not considered to be alive. Now, that seems very strange to people when you tell them that, because they think, well, wait a minute, what do you mean it's not alive? It infects my cells, it infects my body, it makes me sick. I know that it gets replicated, it gets reproduced. I know it can be transmitted to other people. How can it not be alive? It's not alive because it's not cellular. It's not made out of the kinds of things that cells are made out of. And therefore, it can't do the functions that cells can do. One of the key functions that all cells can do is reproduce themselves. All living cells have the ability to divide, at least at some point in their life cycle. Viruses can't do that. Viruses can't divide. So when a viral particle comes into a human cell, the question is, how does more viral particles get made? And the answer is, the virus doesn't do it, the cell does it. Viral particles are considered parasites, intracellular parasites within a cell. They have to go. The cell will then make copies of the virus. The cell has no way to know that that viral material is somehow dangerous to it. So the cell does what cells do, which is copy the genetic material inside the virus and make new viral particles. So yes, a cell is an important part of the biological hierarchy because that structure, the cell, is the subunit of all living things on our planet. Now, single-celled living things, their hierarchy would end right here. Remember, we said there are some single-celled organisms that are prokaryotes. And prokaryotes don't have any organelles. So for them, their hierarchy would be atoms joining together to make molecules, which come together to make cells. And that would be where their hierarchy ends. But you and I are different. You and I are eukaryotic, and we are multicellular. So our hierarchy is more extensive. We have atoms joining together to make molecules, which join together to produce organelles, which join together to make cells. And cells can come together to create what we call tissue. Now, all tissue is, by definition, all tissue is a collection of cells that are all working together to do a similar list of functions. And those cells are brought together and held together with a material that's called extracellular matrix. Extracellular matrix is what holds cells together and creates a tissue. You have 4 different kinds of tissue in your body. 4 different kinds of tissue: cells and extracellular matrix coming together to all perform a particular list of functions. Does anybody know the name of some of our tissue types? What kind of tissue do you find in human bodies? Any idea?
Adipose.
Adipose, that's a great answer. Adipose is actually a subtype. Adipose is actually a type of connective tissue. Good job. So connective tissue is one of our tissues.
Muscle.
Anybody know what's the name of the tissue that lines cavities in our body and lines the surface of our skin?
Epithelial.
Epithelial, good. Epithelial tissue, connective tissue. There are 2 other kinds. Any guesses?
Pseudostratified.
Pseudostratified is a subtype of
Okay.
epithelial, good.
Muscle tissue.
Muscle tissue, very good. And there's one more.
Is it nerve tissue?
Yes,
Or,
very good. Nervous or
and
nerve.
do you think you could write down a list for the spelling of the tissue?
Oh, absolutely. In fact, I'm going to do that right now. So next to this word tissues, I'm going to write down, whoops. It really wants me to make a list here. Epithelial, connective, muscle, and nervous. 4 kinds of tissue. That usually surprises people when you tell them that. They would think that we have many more kinds of tissue than that. The truth is there are a lot of subtypes of tissue. There are different types of epithelial tissue. There are different types of connective tissue, different types of muscle tissue. So yeah, there's a lot of subtypes of tissue in our body, but all of them belong to only one of 4 groups. A group of cells and extracellular matrix, creating a unit, a group that's going to do a similar job for the body. Now, the next most complex level in the hierarchy is organs. When tissues, different tissues come together, again, to perform a similar function, we refer to that as an organ. So for example, epithelial tissue and connective tissue can come together, does come together to make a lot of our organs. Muscle tissue is often found within organs as well. When organs come together to perform a particular function. We refer to that as an organ system. Now, you've heard of organ systems before, for example, the respiratory system or the cardiovascular system, the urogenital system, the musculoskeletal system. We have lots of systems. And within a system, there are different organs. They're all, again, working together to perform a particular list of functions. The last level, therefore, in the hierarchy is the organism. When you bring multiple organ systems together, you create an organism, such as a human being. All right. So again, it's important that we know these types of terms because you'll hear me refer to things during the semester while we're together. You'll hear me talking about organisms. You'll hear me talking about tissues. You'll hear me talking about molecules. And it's important that you have a sense of where that structure fits in this sort of a hierarchy of structures. Now, if you were to take other types of courses, they might make this hierarchy go even longer, even wider, if you will. For example, when we bring many different organisms together, we can create populations, and different populations can create communities, and different communities can come together and create ecosystems. So our biological hierarchy tends to end with organism, but there are certainly other levels in the hierarchy that can be described. I mentioned a few minutes ago that all living things are made out of cells. Life on our planet is cellular. We can go further than that by listing the things that cells have in common. There are lots and lots and lots of different cells on our planet among all the different organisms, but they all share a certain list. They share a list of structure and they share a list of function. On this slide, we're looking at some of those functions. Living things, as we said, are always made out of cells, either one or more cells. Living things have the ability to reproduce themselves. Cells have the ability, at least at some point in their lifespan, of dividing and producing more cells. Organisms, entire organisms, have the ability to reproduce. Living things have the ability to obtain and use energy. Living things can maintain homeostasis. Remember, homeostasis is a term that refers to maintaining a certain state within the body. In our human body, for example, there are lots and lots of organ systems that are combining to do all kinds of work to keep us alive every day. And within those systems, there are certain variables that have to be held within a normal range. Your temperature, for example, your blood pressure, your blood sugar, your water balance, all those kinds of things have to be maintained within a certain fairly narrow normal range. The processes that allow that to happen are what allow for this homeostasis to be established. Living things have the ability to respond to stimuli. That's a really important one. Cells are surrounded by a membrane, and tucked into that membrane are what are called receptors. Now, there are many, many different kinds of receptors, but they share something in common. They're made out of protein. and they have a certain shape. All kinds of things that interact with cells, all different kinds of molecules that come in contact with cells, sometimes bind to those receptors. And in that way, the cell is receiving information about its environment. It's receiving stimuli from the environment. Only living things are able to do this. Finally, living things grow and develop or change throughout their lifespan. Now, when we're talking about a single-celled organism, the organism is only one cell. Growth means division. For example, when bacteria grow, bacteria are dividing to make more bacteria. When you and I grow, we physically get more and more cells as well. That makes us taller and wider and weigh more and so on. The other thing that living things do is they develop or they change over time. Living things don't stay exactly the same from the moment that the organism comes into being till its death. It tends to change throughout that time period. If we go back to viral particles for a minute, and I'll use the COVID virus as our example, viral particles don't meet those functional definitions for living things. Viral particles, for example, don't grow or change. Viral particles can't respond to their environment. They can't receive stimuli. They don't have receptors. Viral particles cannot reproduce themselves. Remember, what they do is they come into cells because they can stick to cells. They can stick to receptors. They come into cells and the cell makes copies of them. So they simply don't meet any of our functional definitions for what a living thing is.
I'm sorry if this is going off on a tangent,
No, that's okay.
but I'm curious, how do viruses find cells if they don't
Oh, that's
respond
a
to
great
stimuli?
question. I actually had that same question in an earlier class today. I think that's such a great question. It's easy for us to imagine that a virus is, what a virus does is purposeful, right? It's easy for us to think that a virus sort of seeks out a cell because the virus somehow has an understanding that if it wants to persist, if it wants to keep going, it needs to get into a cell so the cell can reproduce it. But the truth of the matter is, the interaction between a viral particle and a cell is just totally random. There are receptors all over the surface of cells, and viral particles must bind to a receptor. When they bind to a receptor, that action will cause the cell to bring the viral particle in, okay? So people want to think that somehow a virus must purposefully look for a receptor to stick to, but in reality, it's just a completely random process. It's just a complete collision. That's all it is. So in a pandemic time, when there's literally billions of people who are all susceptible to one viral particle, what happens is, I mean, quadrillions, I don't even have a number big enough. The virus gets into so many people and into so many cells that there is just a ton of viral particles in the environment. And as we know from this particular virus, the SARS-CoV-2 virus, what happens is you just get unlucky. You breathe in particles that happen to be in the air around you. You breathe them in. Those particles, just like all particles in the air, dust particles, for example, they're just going to collide with the cells in your reproductive tract. And every once in a while, your respiratory tract. And every once in a while, the viral particle will collide with the appropriate receptor. And when that happens, when that accidental collision happens, the particle and the receptor will bind to each other, and the virus will be brought into the cell. The more people are susceptible to the virus, the more viral particles are out in the environment, and the greater the chance that you're going to breathe them in. And the more particles you breathe in, the greater the chance, the greater the probability that the particle will collide with the receptor, and you'll get infected. Part of the reason that we work so hard in public health to get people vaccinated is because vaccinated people have antibodies against virus. And even if the viral particle gets into your cells and your cells start to replicate it, your immune system is ready. And as more viral particles get produced in your cells, the antibodies will find them, stick to them, and kill them, destroy them. So yeah, there's no purpose behind a viral particle finding a receptor or meeting a receptor. It's just a random collision in our airways. It's a little bit scary to think of it that way, but that's absolutely the truth. It's just a random event. It's just something that involves spinning air currents, bringing viral particles in contact with the cells that are lining your lungs. Good question. When we look back into the history of biology, into the work of early scientists in biology, there are a couple of names that we should be familiar with. And those are the people that first helped us understand that living things are made out of cells. There was a scientist named Robert Hooke, who did some work in the mid 1600s using a very, very primitive microscope. This is actually a drawing of the microscope that Hooke created for himself. Now, compared to the microscopes we use today, it was very, very primitive. It could only magnify things about 30 times. But, you know, for his time, Hooke was, he was doing something revolutionary. He was examining very, very small things that were not visible to the naked eye. So what Hooke did in his most famous experiment was he took a piece of bark off of a cork tree and he examined it underneath his microscope. Now, if you look at this image on the bottom of the slide, this is what cork bark looks like under the microscope. Now, obviously, a cork tree is it's a plant, and the cells of plants tend to be fairly large. So what we're looking at here are just lots and lots of cells in that bark tissue. Now, when Hooke saw these units, these cavities or compartments of this plant tissue, he had no name for them because no one had described them yet. And what he called them was cells. And he got that word from his culture, from the culture of that time period. You see, during Hooke's day, it was very common for young men to decide to join the priesthood. And what they would do is go to a local monastery and become a monk while they studied. And in those monasteries, every monk had their own quarters. They had their own room. It was usually very sparsely furnished. It usually had a bed and maybe a chair or a desk, and that was it. And they referred to them as cells. So a monk would go to his cell at night to go to sleep. So Hooke knew that term for that reason. And when he looked at this sample of cork bark, that's what came to his mind. He thought about the rooms or the cells inside of monasteries, and he decided to call these little compartments cells. We still use that name today. Now, there were lots of other scientists who did work around Hooke's time. And through the rest of the 1600s and into the 17 and 1800s, all of these scientists sort of came to a consensus that we refer to now as the cell theory. The cell theory was first published in 1830. So there's no one scientist who gets credit for coming up with the cell theory, but we still use the cell theory to this day. It still holds to this day. The cell theory tells us two things. Number one, that all living things are made of cells. And number two, that all cells come from pre-existing cells. In other words, you can't just create a cell out of thin air. A cell has to come from another cell. One cell has to go through a process of cell division in order to produce new cells. All cells come from other cells, come from previously existing cells. Now, the way cells divide really varies. It depends on the organism that you're looking at. Generally, cell division can be described as being either asexual or sexual. So you and I obviously use sexual reproduction in order to make more of our cells, to make more of our organism. In sexual reproduction, we're bringing a male version of the organism together with a female version of the organism. And they're going to combine what are called gametes. Now, the word gamete is sort of the scientific term for a sex cell, a sperm cell, or an egg cell. In sexually reproducing organisms, the two gametes, the female gamete and the male gamete, have to come together. And those two create a new individual. We refer to a single-celled human being as an embryo. So an embryo is a brand new organism. It's a combination of genetic material from its mother and genetic material from its father. What's important to remember about sexual reproduction is this. First of all, most organisms on our planet do not use sexual reproduction. Most organisms use asexual reproduction. So we're kind of in the minority there. The other thing to remember is this. Sexual reproduction produces offspring, children or offspring, that are genetically unique. They are not simply copies of their parents. That's a really important thing to know. There is only one of you. You are unique in all of the earth. And that's because every gamete is unique. So if you're a female, you have many, many, many eggs that you will produce in your adult lifetime. If you're a male, you'll have many, many, many sperm that you'll produce. But each one is unique in terms of the genetic material that it contains. Therefore, with every unique egg and every unique sperm, every embryo that's produced will be unique. Sometimes people will ask me, well, wait a minute, how can we get identical twins then? Well, the answer is that what happens in identical twinning is that embryo that has a unique combination of DNA in it, it's actually going to split early in development and create two embryos that are identical. But they came from one original embryo. So sexual reproduction does something very important evolutionarily speaking. It creates a whole lot of genetic diversity. All of the human beings walking around on planet Earth, with the exception of those identical twins, they're all genetically unique. They all have their own combination of genes and versions of genes. That's very useful for an organism in terms of evolution. Most things, though, most living things do not reproduce sexually. Most of them reproduce asexually. And in asexual reproduction, the offspring are genetically identical to the parent. Now, in asexual reproduction, there are actually different mechanisms. So when we talk about asexual reproduction, we're talking about a parent cell dividing to produce offspring cells, or what we call daughter cells. Those daughter cells are going to be genetically identical to the parent. There is no bringing together of two different organisms here. It's one cell dividing. And so the offspring are identical. They're clones, in other words, of the parent. That actually is a very efficient way to reproduce. It takes less energy to reproduce this way. It's a simpler way to reproduce, but it has a problem, and that's that all of the offspring cells are identical to the parent cell. That does not produce genetic diversity, and that can cause problems for the organism in the long term. Over here on the right side of the slide, you're just looking at some simple diagrams of cells reproducing. They're showing that this parent cell with its genetic material shown in blue has to first copy its genetic material, and then it can divide and produce two new cells. We need each new cell that is produced to have its own genetic material, to have its own DNA. So anytime a cell gets ready to divide, whether it's using asexual or sexual methods, its genetic material has to be copied. We need the new cells to have their own DNA. Down here in this lower image, we're looking at one of the types of asexual reproduction called budding. Budding is a process that yeast cells use, for example. Over here on the left, you're looking at a parent cell, a yeast cell. And this dark area right here is the nucleus. And what's happening when it's time to reproduce is a little bit of the membrane is starting to sort of pouch outward. Can you see that? And that's going to create what's called a bud. And this bud has its own copy of the DNA molecule. It's going to eventually separate from the parent cell, and then it's going to start to grow. It's going to get larger until it is a fully formed new cell, offspring cell. Over here, you can see cells that are all budding. Lots of new cells are being produced. So asexual reproduction makes new cells, new organisms. Sexual reproduction makes new cells, new organisms, but they're two very different processes. We said that living things have the ability to obtain and use energy. Now we're going to talk about energy separately a little bit later in the course. But for right now, just remember that when we use that word energy, we're talking about the ability to do some kind of work. So when we say that a cell is using energy, we're really saying that it's doing some kind of work. In physics, the first law of thermodynamics tells us that energy can't be created out of thin air. it also can't be destroyed. The only thing that can happen to energy is that it can be altered in its form. That's a really important thing for us to remember in biology too. Cells can't just create energy, and cells can't destroy or use up energy. Can't happen. It's against the laws, the physical laws of the universe. But what they can do is change it into a different form. And there are several forms of energy that we see in cells. For example, we see a lot of stored energy or potential energy inside of cells. Cells have the ability to use kinetic energy or movement energy. They have the ability to use thermal energy or heat energy. So energy takes different forms in a living cell, but it can't be created just because the cell needs it. Another one I didn't mention was electromagnetic energy. Remember, light has energy, radiation has energy. There are cells that can capture light energy, right? Cells that are photosynthetic, green plant cells and certain kinds of bacterial cells and algae. They have the ability to capture the energy that's in light and use it for their own needs. There's another kind of energy that's especially important in human beings, and that's electrical energy, electrical current. Our nervous system and our muscle tissue makes extensive use of electrical energy. Later on, when we talk about energy in a little more detail, we'll really focus on the difference between potential energy or stored energy and what's called free energy, or the energy that is available for the cell to do work. So all cells have the need for energy. All cells need to do work to stay alive. But the way that they obtain that energy is going to vary. When I talk about obtaining energy, I'm really talking about essentially, where do they get their food? That's a good way to think about it. I have some terms here that I want us to be familiar with. The first one is chemotrophy. The second one is lithotrophy. And the third one is phototrophy. When we talk about chemotrophy, or we talk about an organism being a chemotroph, what we're saying is that that organism gets its energy by eating certain compounds and then breaking down chemical bonds. So for example, you and I use a molecule called glucose for most of our energy needs. So we have to eat it. We have to eat this molecule. We can't just make it ourselves. And then we break the bonds inside of that glucose molecule in order to then obtain energy. We are chemotrophs. We use chemotrophy to get our energy. We break down bonds in other molecules. Now, lithotrophy also involves breaking down chemical bonds. But what a lithotroph does is break down chemical bonds from inorganic sources. Now, when I use that word organic, I'm talking about the scientific term, not the sort of popular term. In science, organic means carbon containing or carbon based. You and I, again, needing glucose for energy. Glucose contains carbon, so it would be considered an organic molecule. You and I can break down organic molecules to get our energy. A lithotroph can break down inorganic material for energy. So things that don't contain carbon. Now, I'm talking about things like sand and rock and other things that you might look at and say, well, that's not a food source. Surely nothing could get, use that as food. But there are actually lots of bacteria and archaea and other more primitive organisms that can make use of inorganic molecules. They can break them down for energy. The last one on the list is phototrophy. A phototroph is an organism that can get their energy from sunlight. So these again are photosynthetic organisms. They're able to capture the energy that is in sunlight, that is inherent in sunlight, and they use that energy to do work. Now, there are a couple other related terms I want us to be familiar with. One is autotroph and one is heterotroph. These relate to those three terms we just talked about. Autotroph, that term translates as self-feeder, autotroph. You are a self-feeder if you are capable of manufacturing the molecules that you're going to break down for energy. In other words, organisms that can do photosynthesis, they make their own glucose. And then they're going to turn around and break down that glucose for energy. You and I are different. You and I are heterotrophs. That word translates as other feeders. We have to consume our glucose. We can't make our own. We have to consume it. We have to eat an external source of the molecules that we're going to then break down for energy. Now, I always mention here, just to be 100% complete, human beings can actually manufacture glucose in our bodies, but we don't do it very well. There is a process, a metabolic process in humans that allows us to build glucose from other molecules. But again, it's not efficient enough. It's not a rapid enough or an easy enough process for us to do all the time. So most of the glucose that we need for energy, we consume. We consume other animals, we consume plants, we consume other organisms in order to get our glucose. That makes us heterotrophs. So humans are chemotrophs and heterotrophs. Next thing on our list was that, or I'll stick with obtain and use energy, but this time I'll talk about how we use it. We obtain it and then we use it, and that's part of being alive. When it comes to using energy, and remember, by energy we mean the ability to do work. There are really only two ways that cells use energy. There are what we call catabolic processes, and there are anabolic processes. Now, what I'm talking about here when I use those terms, catabolic and anabolic, I'm talking about metabolism. Sometimes that term metabolism seems very difficult to understand. And I think sometimes students think that it's a very complicated term. But in reality, all metabolism is the combination of all of the chemical reactions that go on in an organism to keep them alive. So all of the different chemical reactions that go on in the human body to help keep it alive are what we refer to as metabolism, our metabolism. So if we take all of those reactions, we can break them down into two groups: catabolic reactions and anabolic reactions. Catabolic reactions are any reactions that break things down. They break the covalent bonds that hold molecules together. And by doing that, energy is released. Anabolic reactions are essentially the opposite. Any anabolic reactions are any chemical reaction that helps build something new. In other words, build covalent bonds, create molecules. That's an anabolic reaction. So yes, metabolism is complicated because there are lots and lots of different chemical reactions that cells use to stay alive. But if we look at all those different reactions, they really can be broken down into those two groups. The kind that build things and the kind that break things down. The kind that require energy and the kind that release energy. Again, we're going to talk a lot more about energy later on in the semester. Next on the list of things that living things can do was maintain homeostasis. Remember, homeostasis, that term just means a state of stability, a stable body condition. And every different organism has a different homeostasis. They have a different state of stability. For animals, like human beings, it includes things like a stable body temperature, again, stable blood pressure, stable blood volume, water balance, blood sugar level, things like that. Your body works very hard to maintain homeostasis, to keep you in a state of stable body condition. I wanted to introduce a little bit more vocabulary here. And this is just related to one variable, and that's temperature, body temperature. The terms I want us to be familiar with are endotherm, ectotherm, homeotherm, and this last one's a little hard. It's poikilotherm. 4 different types of organism based on how they maintain their body temperature. For example, an organism is called an endotherm if it maintains its body temperature using internal processes. You and I are endotherms. In other words, things happen inside of our body in order to maintain a relatively stable body temperature. We have a normal range for our body temperature. It actually is between 97 and 99 degrees Fahrenheit. That's the normal range for body temperature in humans. And it's all the processes that go on inside of our body that helps us maintain that temperature range. That makes us endotherms by definition. Ectotherms are a little different. Ectotherms are organisms that maintain their body temperatures using the external environment. So that's a very different means of keeping the body at a certain temperature. Here in New Hampshire, it's not uncommon at certain times of the year, if you're driving along roadways, to see certain types of organisms laying on the asphalt. You may or may not have seen a snake, for example, curled up on the asphalt in the morning on the side of a highway. What that animal is doing is it's trying to warm itself up, because during the day, asphalt absorbs a lot of heat. It absorbs heat from the sun, it stays warm through the cold hours of the night. And these ectotherms, like snakes, will come and they'll lay on the asphalt because they use the external environment to keep their body warm. They'll lay on the asphalt so they can absorb the heat from the asphalt and warm themselves up during the day. Those are ectotherms. Now, a homeotherm is a little bit different. Hold on just a sec. Let me try that one more time. A homeotherm, in comparison to the other two types, a homeotherm is simply a term that describes any organism that can maintain a constant body temperature. Homeo meaning, or homo meaning same. So you and I are also homeotherms. We are endotherms. That's how we keep our body at a constant temperature. And we are homeotherms because we do maintain a constant body temperature. Endotherm, homeotherm. Poikilotherms, that's a horrible word to try to spell, by the way. Poikilotherms are simply organisms whose body temperature varies according to the environment they're in. So again, my snake story, that snake's body temperature is going to be very different depending on what kind of an environment it's in. It's going to experience different body temperatures based upon what's happening in the environment. If you put a snake, for example, into a cold environment, its body temperature is going to drop way down. If you put a human being into a cold environment, at least within reason, our body temperature is still going to stay the same, right? You and I can be out in the cold New Hampshire air during the winter, but keep our body temperature at 97 and 99 because you and I are homeotherms. A snake can't do that. A poikilotherm doesn't do that. A poikilotherm's body temperature really varies. It just depends on what the temperature is outside. And they suffer consequences from that, as you might imagine. That snake I mentioned, a cold snake is a snake that can't move, really. They can't move through their environment because they're simply not warm enough to do that. So snakes are very vulnerable when they get cold. Poikilotherms get very vulnerable when they get cold because they don't have enough body heat in order to do the things they need to do, like move, get away from predators and things like that.
What would like getting a fever be referred to as then? Because
Oh, that's a great question.
at that point you aren't maintaining like your own body
So
temperature.
that's exactly right. So our body works really hard at maintaining body temperature within the normal range. We have structures in our skin called, well, there's a couple of different things. We have blood vessels in our skin. We have sweat glands in our skin that are involved in maintaining body temperature. But when we talk about our normal range, we're talking about our core body temperature, right? We're talking about the very core temperature of the human body. And the body works very, very hard to keep it in the normal range, keep it at that homeostasis condition. Now, when a human develops a fever, what's happening is purposefully, the temperature is allowed to go out of the normal range. So the mechanisms that keep us in a normal range are essentially overridden temporarily. You have to remember that fever is an immune response in us. So it's something, it doesn't happen to us. It's something that we make happen. That seems weird, I know. But fever is actually a way that the human body and other animal bodies fight an invader, something abnormal that has gained entrance into the body. During inflammation, the body temperature rises because immune cells are releasing chemicals that essentially override those processes that keep us at a normal range. The body heats up And that heat is used to kill whatever it is that has invaded, a bacteria, a virus, a fungal infection, all different kinds of things. So a fever is actually a purposeful exit out of normal range for a while, for a certain amount of time. Now, it's also true that sometimes fevers get out of hand. Sometimes the immune system overreacts to infection, especially when we're very young and our immune system is still sort of learning how to do things. Sometimes fevers can get out of hand, and young children, for example, often will go up to 103, even 104. And at that point, the body can actually get into trouble because the fever can get high enough that it not only damages the invading organisms, it not only kills the invading organisms, but it can start to damage normal cells. And that's why fevers can become medical emergencies. Sometimes we have to take steps to bring a fever back down. Does anybody happen to know, just out of curiosity, does anybody happen to know what the danger temperature is in a human where it becomes a medical emergency?
103.
103. Type it into chat if you have a guess. We have 103. I've got 104. Well, it's going up. 105. Ooh, 115. That's a high one. 231. That's a good one, Elijah. That's a little high. It's actually right around 105, 106. It's considered a medical emergency at that point. Here's what I mean by a medical emergency. If you came to an emergency room and you had a temperature, a body temperature of 105, they're going to take immediate steps to bring your body temperature down. In other words, they're going to start giving you a cold intravenous fluid. They're going to start putting ice packs around you. They're going to do what they can to quickly bring your body temperature down. If you go into an emergency room with 102 or 103, don't get me wrong, they're still going to try to give you some medicine to help your fever get better. But if you come in at 105, you're in a state where you could die. You could die if that temperature gets much higher. So that becomes an emergency. And the reason for that is because of your cells. Our cells are surrounded by membrane. Every single cell is surrounded by its own membrane. And that membrane is vulnerable to high temperatures and very low temperatures. What happens at a very high temperature is the particles, the molecules that make up the membrane begin to vibrate at such a rapid speed, such a velocity that the membrane literally falls apart. It cannot do what it needs to do to keep the cell alive. And at high temperature, your cells will begin to die because the membranes start to malfunction. Okay, so here's the opposite end of the question. At what temperature, at what low temperature, so hypothermia, at what low core body temperature does it become a medical emergency? Any ideas? Remember, we're talking about your core. We're not talking about, cold arms or cold fingers. We're talking about your core temperature. I've got a 93, 83, 91, 89. Good guesses. These are good guesses. It's actually right around 88 Fahrenheit. This is a strange little trivia thing for you to know. When temperatures go down too far, there's another effect on the membrane. Now, when I talk about vibrating molecules, remember from chemistry, remember from your chemistry classes, all molecules move, all molecules vibrate, even the molecules in your desk or your notebook. are moving. We can't see them moving. We can't experience them moving, but they all have movement down to absolute zero, right? The higher the temperature, the more the movement, the more the vibration. So in a cell, as you start to get down to about 88 degrees, the movement of the molecules in your membranes becomes so slow that the membrane essentially solidifies. And when a membrane solidifies, one of the things that it can no longer do is conduct electricity. You need a nice soft fluid membrane to conduct electrical current. So what happens when the body gets down to about 88 is the heart stops. Think about what your heart does. Your heart sends out an electrical signal, 60 times a minute to cause the heart to contract and pump blood. So when people get down to about 88, they go into cardiac arrest. Just another little trivia question for you. So we have a range of temperature from about, again, 88 up to about 105, where we've got to stay or we won't survive. And most organisms have that kind of a limit to what kind of temperatures they can endure. So just some terminology for us today that we're learning. The next thing on our list that all living things can do is they're able to pass hereditary traits down to their offspring. Remember, all living things on our planet, at least, use DNA as their genetic material. All organisms use DNA. And inside that DNA, there is code. There are genes that contain code that is then passed on to the next cells. When that organism divides or reproduces, it passes that genetic material on to the offspring. Now, we pass the material down in different ways because cells divide in different ways. I have some terms here I don't want you to worry about today. Bacteria and archaea are two kinds of single-celled organisms, prokaryotic organisms. They don't have any organelles. So their DNA molecule is actually floating around in the cytoplasm, in the cell. These are single-celled organisms. And their DNA molecule is actually in a structure called a nucleoid. It's floating in the cytoplasm. That's very different from our cells. You and I belong in the group called eukarya. We are eukaryotes. Our DNA molecule is kept in a special compartment in our cells called the nucleus. And our DNA is in a form called chromatin inside that nucleus. So there are differences between us in terms of how we house our DNA, but we all have to pass it down from one generation of cells to the next. This is the point where I also want to introduce something called the central dogma. There is a dogma in biology. There's something that is sort of at the very base, the very foundation of all the ways that we understand life. The central dogma is a series of statements. It's 3 statements in total that tells us how information flows from that DNA molecule, keeping cells alive. The first statement of the central dogma is that DNA is transcribed to make RNA. I have that word transcribed in green because the other term you'll hear is transcription. Transcription is the name of the process. DNA is used for a process called transcription. And what transcription does is build RNA from that DNA. The second part of the dogma tells us that the RNA will then be translated. In other words, it is used for a process called translation. RNA is translated to build protein. And finally, the third statement in the dogma tells us that it's proteins that give cells their traits. It's proteins that are really at the core of what a cell is structurally and functionally. So DNA is transcribed to make RNA. RNA is translated to make protein, and it's protein that gives cells their traits. We're going to refer back to the central dogma a lot because it really is central to our understanding of how cells work. This picture over here in the corner of the slide just represents the dogma. Up at the top, this drawing shows a molecule of DNA. It's a double-stranded molecule, what we call a double helix. And the drawing is showing us that this DNA is going to go through a process called transcription. And through that process, an RNA molecule will be built. This is a particular form of RNA that we're talking about, but all RNA is made through transcription. Notice that the RNA, this type of RNA anyway, is shown as a single strand, not a double strand. This single-stranded RNA molecule is then going to be put through this process called translation in order to create proteins. Now, sometimes when I first introduce the central dogma to students in this class, they say to me, well, it's not just proteins that give cells their characteristics, right? I mean, there are other macromolecules inside of cells. There's protein, there's carbohydrate, there's lipids. And don't all three of those give cells their traits or their characteristics? And the answer is yes. But in order to make carbohydrate, in order to make lipid, you need enzymes. And enzymes are made out of protein. So it really is protein that gives a cell its traits. Proteins are used for structural reasons, and proteins are used for functional reasons, enzymes. Enzymes are made out of protein. And it's those enzymes that help build all the other molecules that are going to give a cell its structural and functional characteristics. The next thing on our list of traits that living things have, we said that they can respond to stimuli. I talked a little bit about the receptors that are found in membranes that surround all cells. When we say that a cell or a living organism can respond to stimuli, there are actually lots of different stimuli. There are what we think of as chemical stimuli. In other words, various chemicals can interact with a cell and the cell can respond. There are electrical stimuli. There are thermal stimuli, heat stimuli. There's light. There's sound. There's touch or pressure stimuli. Different cells can respond to a whole list of different kinds of stimuli. And the way a cell perceives a stimulus, again, is through these things called receptors. Receptors are just proteins again, or protein containing molecules. And they are specifically built in order to bind to certain materials, certain chemicals, and so on. They're found both on the surface of the cell and also within a cell. There are some receptors that are actually found in the cytoplasm of a cell. And that might seem strange at first, but remember, there are some chemicals that can pass right through a membrane and come into a cell. And so they're going to bind to receptors that are actually floating around in the cytoplasm. The key thing is that once that thing, whatever it is, binds to a receptor, something is going to happen. It's the action of that molecule binding to a receptor that is going to cause something to happen inside the cell. It is perceiving information when something binds to its receptor. All right, here's like the winning trivia question of the day. Does anybody know, just by any chance, does anybody know the name of the receptor that the SARS-CoV-2 virus binds to? Any idea? Anybody ever heard this? This is real trivia. The SARS-CoV-2 virus binds to a receptor called ACE2, ACE2. Now that ACE term means something. It's an abbreviation, but that receptor normally in a normal human body, a healthy human body, that receptor binds with a certain type of chemical that we have in our body. And it's actually involved in regulating our blood vessel diameter. That's what it does. That's what the receptor normally binds with. And when the thing binds, the blood vessels change their diameter in response. So here's this little viral particle that just happens to be shaped the same way that that normal human chemical is shaped. So that virus can bind to the ACE2 receptor in our blood vessels. When you tell people this, it's a little surprising because we think about COVID as being a respiratory disease, right? That's what we all think of anyway, because those are the kinds of symptoms you get. You get a bad cold, essentially you get, congestion and a cough and a fever, and, people get all kinds of typically respiratory symptoms. But in reality, COVID is not a respiratory disease. COVID is a vascular disease because it binds to receptors that line our blood vessels. You've probably heard that there are some instances of patients with COVID getting heart problems, cardiac inflammation. Some people who get COVID report terrible headaches. Some people who get COVID talk about feeling very confused, getting a brain fog. There have been people with COVID who have gone on to develop diabetes. Oof. And there, unfortunately, have been a lot of people with COVID who've gone on to develop just a lasting illness that we call long COVID. All of those problems are related to inflammation of the blood vessels in different parts of the body. So COVID is actually a vascular virus. It infects the cells that line blood vessels. So yeah, it does come in through your respiratory tract, but the cells that it invades are actually blood vessel cells, the cells that line your blood vessels. All right. The very last thing on our list of functions that all living things have was growth and development. We said that living things, living cells, are going to change throughout their lifespan. They're going to grow, develop, change in some way. Everything that is alive has a certain time to be alive. They have a certain limited lifespan. And depending on the organism, they may actually have different stages in their life cycle. For example, if you look at a human or other types of animals, we can describe things like the embryonic life stage, the fetal life stage. There's a time that we refer to as being an infant or a child or an adult, just different stages in our life where we are very different organisms. We look different and we function differently. Even outside of these more obvious stages, humans have stages like pre-puberty or puberty. We have a reproductive stage to our life. If you're female, you have a stage where you can become pregnant or you can lactate to feed an infant. There's even a stage later in life where we stop reproducing, right? Now that stage is more obvious in females. Females go through a process called menopause, where they stop releasing eggs for fertilization. This term senescence refers to the end of the lifespan. So we have a reproductive senescence. We have a time where we stop reproducing, if we're female anyway. The truth is, male animals can actually reproduce throughout their lifespan. They don't reproduce as well when they get older and older and older. Their sperm quality goes down the older they get, but males actually make and release sperm throughout their lives. But we females are a little different. Females have a limited lifespan for reproduction. So let me ask here, if anybody has any questions about these functional characteristics that living things have, this list that we've gone through today. Does anybody have any questions about anything that we've talked about? It's really interesting when you start to think about what it means to be alive. It's an interesting question. And again, it's important to point out that these definitions and these ideas we have about life are based upon our existence here on Earth. We don't know if there is life on other planets. We don't know because we can't know, at least not yet. But it's interesting to note that recently, anyway, in space travel, as we've gone and collected samples from Mars, for example, we have good evidence now that there was a time in the history of Mars when there may well have been life on that planet. And we can say that because we have found evidence of water on that planet. So everything that we know about what it is to be alive is based upon our experience here on our planet. And who knows, there may be a time in the future when we discover life elsewhere. And that life may have very different characteristics than ours does. Just something to think about. I want to switch gears now, and for the last few minutes that we're together, I want to talk a little bit about scientific study. We'll bring this up again and again through the semester as we do our lab work. But I just wanted to introduce a couple of concepts today about scientific study, just to make sure that everybody has the same sort of foundational knowledge about this. We talk a lot in this course in lab about research studies and the different kinds of research studies. So it's important to understand that whenever we're doing experimental work in science, we're really doing one of two kinds of experimental work. There's observational science and there's experimental science. And those are two very different things. Observational science is no less important than experimental science. It's just conducted differently. An observational type of experiment is one where we are observing, as the name would suggest. We're observing and we're describing the subjects or the conditions without actually interfering with them, without manipulating them or interfering with them somehow. That's what we call observational science. Experimental science is very different. In experimental science, we are purposefully interfering. We are manipulating the subjects or the conditions of the experiment to see what happens. And that's where you get terms like experimental or treatment groups versus control groups. You've probably heard that every experiment, every experimental study has to have what we call a control group, which is a group that is there for comparison purposes. The treatment group is the group that is being manipulated, the group that something is happening to. And the control group is identical in all ways to the treatment group, except for that specific manipulation or treatment that we're doing. So for example, let's say we're doing a study, we want to study a new drug. We want to find out, let's say, whether or not this drug can lower your blood pressure. Let's say that's why the drug is being studied. This is a drug that, that this company hopes to sell as a blood pressure lowering drug. So if we're going to design an experimental study, we're going to need a treatment group and a control group. The treatment group is the group we are going to manipulate. We are going to treat. In this example, we're going to give them the drug and we're going to see what it does to their blood pressure. The control group is there as a comparison for us. The control group is there so that we can see a comparison in a very similar group of test subjects that aren't given the treatment, that aren't given the drug. We try to match them. We try to match our treatment group and our control group. We want them to be generally the same sex, approximately the same age. We want them to have approximately the same general health. We want to know that they are very similar in all things. One group gets the drug, the other group doesn't. And then we know at the end of the study if what's happening in the treatment group is due to the drug, or if it just would have happened anyway. We look to see that what's happening in the treatment group does not happen in the control group. If it happens in the control group too, then you can't attribute it to the drug. Now, that's a very simple way to explain experimental studies, but you get the gist of it. We put a control group in because we want a comparison. We want to know that if all other things are equal, would this drug still have had an effect? Now, there's a couple of other terms just to be familiar with, and this is about reasoning. Whenever we perform experiments, we have to come up with the experimental design. We have to come up with how we're going to conduct this experiment in order to test our hypothesis. Generally speaking, there are two categories or types of scientific reasoning, what we call inductive reasoning and what we call deductive reasoning. In inductive reasoning, where it's defined as a form of logical thinking that uses observations to arrive at a general conclusion. Now, what do I mean by that? I mean that we're going to look at what's happening and we're going to decide that what's happening has caused this or has led to this. Inductive reasoning is the kind of reasoning that is commonly used in those observational types of studies. Now, remember, just because it's observational doesn't mean it's not good. We need observational research studies. They give us a lot of information. Observations, remember, they can be qualitative or quantitative. What's the difference there? What do I mean when I talk about a qualitative versus a quantitative observation? Can
Well,
anybody go ahead?
Quantitative usually in high school referred to like a quantity. So like you're measuring something, like a difference between two things, whereas qualitative is like, oh, in this reaction, there was a color change or like something like that.
You can literally see what it means in the terms, right? It's quality versus quantity. So when we're talking about a qualitative observation, it's like you said, it's descriptive. It's talking about the quality of something. Did it change color? Did it grow? Did it move? Did it? Quantitative means quantity. It means some number, something that we are measuring using numbers, using units. Okay, so we might be measuring temperature, or we might be measuring length, or we might be measuring volume or pressure or blood pressure. Quantity, quantitative, involving those numerical measurements, and qualitative being the quality of something, an observational quality about something. Good. Now, deductive reasoning is different. In deductive reasoning, we're going to use a general principle or some other natural law, essentially, in order to forecast results. All right, now you'll understand this in just a minute. I'll give you an example. But this kind of reasoning is actually more common in experimental science. Again, experimental studies can be either quantitative or qualitative. They are no better or worse than observational studies. They're just different. So we have to think about scientific method when we talk about quantitative versus qualitative. When we talk about inductive versus deductive reasoning. Remember, the scientific method is quite old. It was first described by Francis Bacon back in the 17th century. Bacon was talking about an approach to science that we could all use every single time that we were doing some kind of experimental work. First thing in science is always observation. We always see something. We see something that catches our eye, that we're interested in, and we want to know something about it. Think about a very basic example. The classroom is too warm. I'm sitting in a classroom and I'm observing that the classroom is too warm. That's a pretty basic observation. From the observation, we go to a question. Why? Why is the classroom so warm? We always have to be asking a question in science, and it's that question that helps us develop or propose a hypothesis. Perhaps it's too warm in here because somebody forgot to turn on the air conditioner. That's a good hypothesis. We need to know exactly what a hypothesis is, and this is an important thing to note today. Put a star next to this. A hypothesis is an educated guess, yes, but it's an educated guess that can be tested. It's not a hypothesis unless you can test it, experimentally. Now, if I make the hypothesis that it's too warm in this classroom because somebody forgot to turn on the AC, I can test that. I can turn on the AC and I can see whether or not the warmth in the classroom changes. Something else to know about hypotheses is this. A hypothesis does not set out to prove anything. That's really important to know. The other thing is, with a hypothesis, we have to set variables and we have to set controls. We have to set variables. We have to know what it is that we're going to be observing or measuring in our experiment. And we have to have something to control that too. That's that control group I mentioned. A lot of times, I think students think that they want to believe that they're proving their hypothesis in their experiment. I'm going to turn the AC on. I'm going to show that the classroom's going to go back down to its normal temperature. Therefore, I have proven my hypothesis that the reason the classroom was too warm is because someone forgot to turn the AC on. We do not prove things in science. What we do is we add our conclusions to a body of information, to what we call the scientific literature. We are adding what we found into what is already known. Science is actually done by consensus, and it's done over time. If I find that such and such, that a drug helps reduce blood pressure, other scientists might study that drug too. They may find that it actually doesn't reduce blood pressure. They may have conflicting results to mine. And the only way we're going to resolve that is by doing more studies. Science is done by consensus. You should never, ever think that one experiment proves anything, because it simply can't. Science is limited by statistical analyses and experimental design. So we always have to be very critical about our findings. We have to be critical of what we find in our experiment, because we don't want to over conclude what it means. So yeah, we don't tend to use that word prove. All right, we're just about out of time here. There's a couple more slides in this set. Actually, there's one more slide in this set, and it's the rest of the scientific method about predicting and experimenting and collecting data and analyzing data and so on. I know you all have heard of the scientific method at one time or another in your career. We'll talk more about methodology and experimental design when we're actually doing our experiments in lab this semester. All right, if no one has any questions, I'm going to let you go, and I will see you again next week. Remember, you don't have any homework to do for me this week, but get that survey into me if you haven't already.
Don't
Okay?
we have like a weekly checkup thing due as well?
Say again?
Don't we have like a weekly checkup due this week? Oh wait, that might, sorry, that's for
That
my,
might be another class, maybe?
that's my other
Okay.
class. Sorry.
That's okay. Believe me, I get confused from one to another too. So yeah. No, all you got to do for me this week is get that survey in. Okay. And I will see you again on Tuesday next week in the classroom. Hooray. All right, everybody. Enjoy the rest of the day.
Thank you.
We'll see you
Thank
next
you.
time.